Next, we have a joint presentation by Joe Costello. He's a professor and director of the training program in translational brain tumor research at the Brain Tumor Center, and by course co-director Nancy N. Oberheim Bush. Their presentation is titled Temozolomide-induced hypermutation mechanisms and clinical relevance. Joe?
Thank you to everyone for being here, and a special thanks to Nancy Ann and Bjorn for this organization. Wonderful symposium. I'll be presenting in conjunction with Nancy Ann on temozolomide-induced hypermutation mechanisms and clinical relevance. I would like to say that none of this work would have been possible without the generous support of Alan and Ashley DeBerry. What Nancy and I hope to address in this presentation is are three questions. What is hypermutation? How does it happen in IDH mutant low-grade glioma patients? And why does it matter to the patient and to their oncologist? We have no disclosures. IDH mutant low-grade glioma in adults, the tumor type that we study, and these are generally very slow-growing tumors where recurrence is sometimes years or even decades later. The primary treatment, of course, is surgical resection of the tumor, and in some cases, that is followed by radiation and chemotherapy. Because the tumors are highly infiltrative, surgical cure is usually not possible, and the tumors will recur at some point. So we embarked on a genomic study to understand how low-grade gliomas evolve over time and how we could use that information to improve therapies. So as I mentioned, low-grade gliomas do recur, but there's a wide range of time between the diagnosis and recurrence. In some cases, the patient will recur with a low-grade tumor again, and often that is clinically manageable. However, many patients will come back with a much higher grade, more aggressive tumor like glioblastoma. That process is called malignant transformation. So we set out to do a genomic study to try to understand the mechanism of malignant transformation, with the idea that we could then use that information to apply existing therapies in a better way or develop new therapies. And in the process of trying to understand that malignant transformation, I think we came up with what may be one of the causes of malignant transformation. So important in this discussion is the fact that some patients will receive the chemotherapeutic agent temozolomide after the initial surgical resection, and that is a strong selective pressure because the drug actually does kill tumor cells very well, but it does change what comes back. This study began quite a while ago, and it's very much a team effort. The entire clinical team here at UCSF, and we had some international collaborators as well. And it was a relatively small cohort, but because we needed to have the DNA from the blood from the patient, the DNA from the original tumor, the newly diagnosed tumor, and a tumor at recurrence. And sometimes, as I mentioned, that could be 10 or more years later. So thankfully, the UCSF Brain Tumor Tissue Bank has collected such samples. So before we discuss the genomic data, I need to talk to you about the chemotherapeutic agent that is sometimes used in these patients called temozolomide, and how does it work? And how is that related to this idea of hypermutation? So temozolomide induces DNA damage at guanines, and that damage can be repaired by a DNA repair protein called MGMT. So in an MGMT unmethylated tumor, temozolomide damage is quickly repaired and the tumor is resistant to cell killing. In contrast, in MGMT methylated cases, the damage is not repaired, and a second repair mechanism kicks in, mismatch repair, but that results in this futile cycling that ultimately will kill the tumor cell. And so this is how temozolomide actually kills glioma cells. However, if this mismatch repair system is also inactivated, this normally cytotoxic agent can in fact be mutagenic. It can induce new mutations, and very specifically a G2A mutation in the tumor. However, we have not really been concerned about this aspect because most low-grade gliomas do not have a mismatch repair defect. They have intact mismatch repair. So I will now show you some of the genomic data from our initial study and subsequent work addressing how the tumor evolves over time, particularly when the patient is receiving temozolomide after the initial resection of the tumor. So here's one patient, patient 18, and we did exome sequencing, sequence all the exomes across all genes in the normal and the tumor and the recurrence. So the initial tumor has about 75 mutations. The recurrence has a dramatic increase in number of mutations, so 2,600, so about 30 times the number of mutations of initial tumor. And this is why we call that hypermutation. It's just an excess of mutations. And 95% of the new mutations that occur are G to A, which is the signature of the temozolomide. So why is that happening if we know that the initial tumor did not have mismatch repair defect? So to cut to the chase here, with our first study on 23 patients, we had 10 of them who had received temozolomide between initial and recurrent tumor. 6 out of 10 had hypermutation. And the more concerning aspect was that all 6 of those patients had recurrences that were now glioblastoma rather than recurring again as a grade 2. This is a small study, and it just raises the hypothesis. It doesn't prove, but raises the hypothesis that hypermutation and malignant transformation are associated. We also wondered whether they could be causally related. And so the panel B that you're seeing on the right side, bottom, illustrates one of the pieces of data that made us suspect that they might be causally related. And that is the 2 pathways that we know are required for a slow-growing low-grade glioma to become high-grade are the AKT-mTOR pathway and the RB cell cycle control pathway. And what we found was that in the hypermutated cases, there were mutations in one or more of these genes. In green represents a G to A mutation, temozolomide signature mutation. They're prevalent in both of these. And if you contrast that to tumors that also underwent malignant transformation, but through a different route, they were not treated with temozolomide, but did so spontaneously, you see a complete lack of these G8 mutations, but you do see alterations in the same genes. So same genes, different mechanism. One case, it is G to A mutations, the other, a variety of different mechanisms. This suggested to us there might be a causal relationship, but it's just a hypothesis at this point. So back to this diagram, I mentioned would this happen if the mismatch repair system is intact and if this model is correct? Well, we noticed that all 6 of our initial hypermutators had in fact acquired a mismatch repair mutation, and those mutations also were G to A, suggesting that the temozolomide initially induced a mutation in mismatch repair, and that created a situation that allowed hypermutation to occur. So we need to take this into a bigger picture, and we have clinical data that I think will help us understand hypermutation and this association with malignant transformation. we can ask, there's this potentially negative outcome, but how long did it take for the hypermutated cell to actually grow? Because we do know that temozolomide is an effective agent in this patient group. And so what this plot shows is starting at time 0, when the patient first received temozolomide, how long did it take before that hypermutated recurrence was found? And the main point is that it's a wide spectrum across the patients, astrocytoma and oligodendroglioma. In some cases, the hypermutation occurred very quickly, more quickly than would be expected in this patient group. But in other patients, it was almost a decade after. This suggests that cells that were hypermutated here either sat there dormant or grew incredibly slowly for a long period of time and eventually manifested as a glioblastoma. So it's a balance between the therapeutic benefit of temozolomide, clear therapeutic benefit, and this potential transformation that is at least associated with temozolomide and may be causally related. So with that, I'm going to turn it over to Nancy Ann to address the question of the clinical relevance of hypermutation in a much larger cohort. So we could not address it in a small cohort, but we now have over 80 patients where we have the genomic data. You can see on this slide how easy it is to distinguish the hypermutators, especially on the bottom panel here, from the non-hypermutators when we use mutation signature, that's that G to A mutation, and number of mutations. So with that, I will stop my sharing and turn it over to Nancy Ann.
Thank you so much, Joe. So yes, basically we have all of this great data that shows that hypermutation happens and how it happens and why it happens, but really what does it mean for our patients? And so we sought out to ask these questions. Is there an association between hypermutation and transformation of a low-grade tumor to a higher grade? What is the prevalence of hypermutation? What are the clinical impacts? And what are the risk factors to develop hypermutation? So we identified 82 patients with initial WHO grade 2 IDH mutant low-grade gliomas, either 1p19q co-deleted oligodendrogliomas or astrocytomas that underwent at least one resection for recurrent or progressive disease and had enough tissue so that we were able to do either UCSF 500 or whole exome sequencing. It's relatively balanced between oligos and astros. The age of diagnosis was slightly older for oligos, which we expect at age 38 compared to 31. And we found that of the 82 patients, 66 had tumors that transformed to higher grade disease. Median time from diagnosis to transformation was 5.9 years for all tumors, 4.8 for astrocytomas, and 7 years for oligodendrogliomas. Now, of these patients in the oligos, 80% of patients did get treated with temozolomide, and about 30% were treated with radiation. In the astrocytoma population, 75% were treated with temozolomide, and about 25% were treated with radiation as well. In the oligos, for the patients that transformed, they all transformed to grade 3 disease. As we know, there's no grade 4 oligodendroglioma, so 80% of patients transformed. In the astrocytomas, also 80% of patients transformed. Pretty equally, 40% transformed to grade 3 disease and 37.5% transformed to grade 4 gliomas. So what did we find? So of the patients that had transformed disease and were previously exposed to temozolomide, 57% of those patients had evidence of hypermutation in their recurrent specimen. This was fairly equally balanced between the oligodendrogliomas, where we saw 61% of patients having evidence of hypermutation, and the astrocytomas, where we saw 53% of patients with evidence of hypermutation. Just noting that 0 patients that were not exposed to temozolomide developed hypermutation, which we expect. We were actually able to validate these results using an independent cohort from the Dana-Farber Cancer Institute, showing very similar amounts of hypermutation in transformed low-grade gliomas that were exposed to temozolomide. Dana-Farber had a little bit less of astrocytomas than we did, and I'm going to return back to that later in the talk. We have a reason for that. So we also found that hypermutation tumors were more likely to be high grade after controlling for tumor subtype. We found that 93.5% of recurrent hypermutated tumors were associated with high-grade transformation in the hypermutated specimen. So there were 2 patients that had hypermutation that recurred at grade 2. 1 patient with oligodendroglioma, 1 patient with an astrocytoma. The patient who recurred at grade 2 with an oligodendroglioma had a very aggressive clinical course, had nodular enhancement within 13 months of reoperation, but never underwent a further reoperation to confirm transformation. And the patient with the grade 2 hypermutated astrocytoma similarly had a very aggressive course. They actually did undergo a reoperation 33 months after completion of Temodar, and that patient had evidence of transformation to higher grade disease. In all other cases, hypermutation was associated with transformation to high grade disease. We also wanted to know is it prognostic? So what we found is that patients who were hypermutated that transformed to a higher grade actually had a worse overall survival. So if you transformed and you were hypermutated, you did worse than patients the same grade that were not hypermutated. We were also able to validate this using the Dana-Farber independent cohort, again showing that hypermutation causes a worse overall prognosis. Interestingly, when we started to look very specifically, we found that hypermutation was actually prognostic specifically in grade 3 disease. So if you transformed to a grade 3, your overall survival was worse if you were hypermutated. However, on the right of your screen, what you can see is if you transformed to grade 4, you had an equally poor prognosis. So once you developed grade 4 disease, hypermutation no longer remained prognostic. And again, this could be validated using the independent cohort from Dana-Farber, showing that these results really are validated. We also were able to do a multivariate analysis, and we looked at multiple different parameters, and we found that both radiation and hypermutation were significant predictors of overall survival. So the patients that did the worst were the ones that had hypermutation and were radiated, and the patients that did best, no radiation, no hypermutation. And this, again, also we could validate with using our independent cohort. All right, so I just told you that hypermutation is a common event. It also, we're showing you that it's associated with high-grade transformation, worse overall prognosis. And when we were looking at these cases, we noticed something really interesting. There were these very distinct patterns of reoccurrence, and that normally when you see progression of a low-grade tumor, it's contiguous with its primary site of disease or the surgical resection cavity. We started to notice that there were many patients that were having recurrence in multiple different locations. So we defined discontiguous disease as tumor separate site from where your original tumor site was. And we found that this was significantly higher in our patients with hypermutation, happening close to 42% of the time compared to only 12% of the time in hypermutated patients. And that this was associated with both tumor grade and then time for transformation. As well as molecular subtype, there was a trend that this happened more commonly in the astrocytomas compared to the oligos, although that did not reach significance. And so we took an even closer look. And in discontiguous disease, we defined it in 3 ways: ependymal dissemination, leptomeningeal dissemination, and parenchymal distinct sites of disease. And we found that patients with hypermutation had a significant increase in ependymal and leptomeningeal dissemination. So 7 patients with ependymal dissemination, 4 with leptomeningeal, and only 2 patients that had parenchymal distinct sites of disease. This was in stark contrast to the 6 patients in the non-hypermutated group that had discontiguous disease. All of them were parenchymal. We didn't see dissemination in any of those patients. And in fact, in the hypermutated group, we saw 4 patients that had spinal dissemination. One of these patients actually underwent a rapid autopsy of the spinal deposit and did exome sequencing there, and it demonstrated a hypermutated clone identified from a previously intracranial hypermutated clone. So what are the risk factors for the development of hypermutation? I've already told you that oligos and astros seem to be pretty similar. It doesn't depend on tumor type. But something that we did find is that radiation seemed to be protective. So only in the astrocytoma population. If you had an astrocytoma and were radiated, you were less likely to develop hypermutation. And this was actually also validated through the Dana-Farber cohort. And I think that was one of the reasons why in the astrocytomas, Dana-Farber actually had less development of hypermutation. Turns out they radiated their astrocytomas less than we did at UCSF. We were wondering if the amount of Temodar made an impact. Didn't seem to. It could be any amount of Temodar. How about the tumor volume? Also didn't seem to make an impact. Joe already hinted on this, but there was no timing. It really was so variable when patients would become hypermutated. It wasn't soon after, it could be years after, and it was seen for both oligos and astros that way. But the one thing that we did find that was predictive of the development of hypermutation was actually MGMT promoter methylation. So Joe's group did a beautiful study looking at 17 different CpG sites on the MGMT promoter in the initial tumor specimen. And they found that if you were methylated, that you were much more likely to go on to become hypermutated at recurrence. And they found that patients that were becoming hypermutated had higher levels of methylation at almost every single one of those CpG sites. And they could actually create a cutoff of 42.5% methylated of the MGMT promoter was going to go on to predict that you would become hypermutated. So in summary, hypermutation was identified in 50% of transformed tumors exposed to temozolomide. So really a common event. It was associated with a higher graded transformation. It was associated with a worse prognosis, specifically when you transform to grade 3 disease. And it was associated with this really distinct pattern of recurrence where you're seeing ependymal and leptomeningeal dissemination. Radiation seemed to be protective, especially in the astrocytoma population, and MGMT promoter methylation level in initial tumors could predict the development of hypermutation status at recurrence. And with that, I'd like to thank the entire group, Joe Costello's lab. It's been really a pleasure to work with everybody there. Yao Yu was a radiation oncologist here who moved to Sloan Kettering, who did a vast majority of this work as well. All of the neuro-oncologists and the imaging analysis blinded was done by Javier Villanueva Meyer. So thank you, everybody.